And again, I'm really happy to be here. And as Nick mentioned, I'm at Stanford in cardiothoracic surgery is the department that I'm in. But I'm actually trained as a bioengineer, and in fact, I'm actually trained specifically as a tissue engineer. And so today, one of the things that I would like to do is really give you a little snippet about some of the research that we do in our lab, as well as to tell you a little bit more about my story and how I came across this field. And hopefully, this will be helpful, maybe useful, and help to give you ideas in terms of a potential career in the STEM area, particularly in research and potentially in teaching, as it has really benefited me in the past. So today we're going to be talking to you about a topic that we study in our lab that's called tissue engineering, and particularly I'm going to talk ultimately about tissue engineering of artificial blood vessels for treating cardiovascular diseases. But before that, let me give you a little bit of the history of how I came to this field. So let's start back from a number of years ago when I was a high school student. In fact, I was a high school sophomore from New York and had really no idea what I really wanted to do. I took freshman and sophomore classes and really had no idea what I wanted to be in the future. And it was almost, I think, a bit of a luck of a draw that summer I applied to a number of different internships, some that were science related, some were working at finance companies, some were in like a museum, and I sort of applied to sort of anything and everything, because I didn't know. But as they turned out, I got accepted to a summer internship program through the New York Junior Academy of Sciences, and they placed me in a lab. So that summer, I did science as an internship, and luckily, I was really lucky that I got placed in a hospital where my mentor, who was a research scientist, was really a great mentor, and essentially taught me everything that I knew. Up until that time, I'd pretty much taken very basic high school biology and really didn't know anything about research. And he really took me under his arms, and that actually wasn't just me. And so this is actually a picture of the first year of our internship. And he took actually about 5 of us into his lab and showed us everything from how to pipette to doing some really sophisticated basic biology experiments, which at that time was completely new to me. But I think the most important thing about that experience was that the mentorship of Dr. Sunny Luke, who was my mentor there at the time, really changed my mindset of what I wanted to do. And at that time, we were doing research related to cancer, ovarian cancer. And at that time, I thought, wow, this is a really great field. Maybe I want to consider being a doctor someday and do research someday. Now, flash forward to what goes on with me now, which is now I'm sort of taking a lot of the opportunities that were given to me back when I was a high school student, and then where the opportunities arise, trying to contribute back, and I think that this is sort of this circle of getting a lot of mentorship while growing up, but then also having the opportunity to give back. So as I mentioned, right after this internship, I was really excited about science. I was really excited specifically about cancer, and I thought, wow, maybe I should be a scientist and study cancer, maybe I should be a doctor. But eventually, I became something else, and which I call a tissue engineer. And on the right is just sort of a famous picture of what tissue engineering is about. Maybe you have seen these pictures, which first popped up in the late 90s when the field of tissue engineering was born, which is essentially creating artificial biological organs that can be ultimately transplanted back into the body for therapeutic purposes. And so this is an example of an engineered ear, which was developed in the 90s, and people thought, wow, it's going to be such a great field. So I'll tell you a little bit about how I went from being someone interested in cancer to then someone who became ultimately a bioengineer and specifically a tissue engineer. So in terms of my training history, I went to MIT, and at that time I thought, wow, they have a really good science program. I should major in biology. And along the way, I discovered engineering as a very practical discipline, which essentially applies, I would say, logical steps to problem solving. And I think that was really attractive to me. Maybe it was personally that I felt that a lot of my pure biology courses, while they were very interesting, were very heavy on memorization, that I felt that, wow, all my engineering classes are open book, and I felt a draw to engineering because it was really about a discipline and an approach to solving problems which can be applied not only to classes but also to everyday life. But ultimately, I was still interested in essentially, biology. So after college, I went to UC Berkeley and got my PhD in bioengineering. And I thought, well, maybe this is a great merger between my interest in biology as well as my interest in engineering, so that I can essentially take both of these two different disciplines to solve biomedical problems, because they're so complex that someone who's purely a biologist may not be able to solve the entire problem, and someone who's purely an engineer cannot solve the problem either. Medical problems are now becoming so complex that you really do need multidisciplinary knowledge to solve these problems. So I thought bioengineering was a good merger between my interest in biology and medicine, as well as applying engineering principles. And after that, I then did more practical research. This is what we call a postdoc at Stanford in cardiovascular medicine. And up until a few years ago, then I started my faculty position at Stanford, as well as at the VA Palo Alto adjacent to that. So I wanted to give you a little bit of a snippet of what does a professor do. Now, granted, there are lots of different professors, and but sometimes as a student, particularly, let's say if you're in college, you may sometimes think that, well, your professors just teach all day and then they grade homework all night, and that's the life of a professor. And although that is a big component of it, many types of professors are out there, but in particular, not all professors only do teaching and homework grading. So I want to tell you a little bit about some of the things that I do. So as a professor, I definitely teach undergraduates as well as graduates, but I'm also a researcher. So I run a laboratory that consists of students as well as postdocs, and the idea is to be able to solve interesting medical and biological questions. And so I think the most interesting thing that I do as a professor, one, is to teach and mentor students, but the other is to think about new and interesting research ideas. And those usually come in the form of writing a grant proposal, which then hopefully, keep your fingers crossed, would get funded so that we can actually have the funds to be able to carry out the experiments needed to answer these interesting biological questions. And of course, along the way, we also do a lot of I would call research dissemination, so which is essentially talking about our research ideas by going to conferences, talking to other colleagues, presenting some of the data at different types of conferences and meetings, as well as giving feedback on other people's grants or research data. So we call that peer review. But this is a little bit of a snippet of what some professors like I do. And although it's not to say that every professor does that, now, why do I want to even get into this field? Why am I in this field? I think that there are definitely a number of perks about being a professor. So for me, I think one of the biggest perks about being a professor is to be able to transfer knowledge to the next generation. In the same way that when I was a high school student and had no idea what I wanted to do, a mentor came along and really opened my eyes. And so I think this is something that I truly believe is life-changing and would love to offer that opportunity to others as well. And so that's what I mean by being in a position to influence the future career of students. And you may know from some of your previous classes, maybe you had a class where, for whatever reason, you didn't understand the material, or maybe the material was not conveyed in an understandable manner, and maybe that class may have turned you away from that subject matter entirely. Like you've X'd out economics, or you X'd out some other type of discipline. Well, it's happened to me too, and I think there's a natural gravitation that way. So being a professor is both a great thing, but it's also a dangerous thing, because you do have the power to influence a student to go to your field as well as to not go into your field. And then third, I put the word flexible in parentheses. So flexible refers to the fact that being a professor does not mean you have a 9-to-5 job, you clock in and you clock out. It's a lot more self-motivated, so you can always work more if you'd like, you can work less if you'd like. So in that sense, we call it a flexible, type of schedule. But I would put that in parentheses, because most of the younger faculty, like I am, tend to work more rather than less. So, as I mentioned before, I have a really strong interest in inspiring the next generation of students, and just want to, tell you some of the things that I'm working on now. So I'm involved in 2 different nonprofit organizations. One is called ISAC, International Institute for Scientific and Academic Collaboration, where we promote youth enrichment and international education through internships, experiential learning. And another one, more recently, called the Science Runway, is as what it says, shining the spotlight on role models in healthcare and life sciences to inspire the next generation of women to join the industry. So this was actually started by a group of high school students, Science Runway. So these are just some of the current ways that I'm hoping, in addition to my current role as a professor, to inspire the next generation. All right, I told you all the great things about being a professor, and it would not be fair if I didn't tell you about some of the stresses, just to be fair. And one of the big stresses is that as a professor where you're running a lab, the lab is kind of like a startup. What does that really mean? Well, you run the lab, so you're like the CEO, you're the CTO, you deal with all the finances, so you're the CFO. You are in charge of hiring all the people in your lab, so you are HR. And you're also responsible for talking about your lab's research at conferences and through other means, so you are PR right there. And believe it or not, you're also the safety officer. In the state of California, the professor is legally responsible for the safety of all of their personnel. So if a personnel gets injured on the job, guess who gets blamed? The professor. So professors become criminally liable for the safety of their staff. So this is just sort of some of the different roles that young professors like I take on. And of course, in order to be able to get sufficient funds to do research, where do we tend to get them? Well, we tend to get them from different federal or state or private agencies, and a very common one is from the National Institutes of Health. So these are just some stats released by the NIH, just giving you a general idea of the percentage of grants that get funded every year. And you can see that there's an overall decline in these percentages with time. And so I think that it is a very tough field currently to be able to thrive in an environment where your funds essentially come from agencies where the money is tight. But I think that there are definitely opportunities, and I think that they are challenges that yet can be surmountable. So, I think the take-home advice about sort of my training history is, for you to think about, well, do you think a research career may be right for you, or maybe being a professor may be right for you? Well, some of the questions that I would maybe ask you to ask yourself is, are you naturally curious about the world and yourself? Do you enjoy searching for answers or knowledge? And then, of course, are you patient and motivated? And I particularly say patient and motivated, because research is more of delayed gratification. You don't get the answer immediately. Sometimes it takes a long time before you actually get the answer. So it does require a certain type of personality in being patient, but still dedicated in your research questions. So anyway, so these are some of the main things that I would suggest if you're interested in a career in research. Okay, so and of course I'm going to start my research portion of the talk, and along the way, if you have any questions, this is very informal. Feel free to raise your hand, and hopefully we can address these questions. And again, there's no particular time limit per se. I just want to be able to share some of the research that we do in our lab. So if we don't finish till the very end of the last slide, it's not a problem. So one of the big diseases that our work is motivated by, particularly within cardiovascular disease, is atherosclerosis. So if you look at a number of different websites, some people may define atherosclerosis as the hardening and narrowing of the arteries due to the buildup of fats, cholesterol, and other substances. So you can imagine if this vessel over here, which is your heart, is essentially clogged by cholesterol and other types of fats, then essentially blood is not going to be able to flow down your heart, and as a result of that, it could lead to a heart attack, something we may call a myocardial infarction, to be more precise. Now, this is the case of the heart. If it happened in the heart, you would have a heart attack, and there would be a decent chance of a fatality there. Now, what if it was a vessel that was not in the heart? And a very common, but also highly undiagnosed disease is called peripheral arterial disease. So it's a form of atherosclerosis where the clogging of the arteries happen in the arms and legs. And PAD affects over 8 million patients, and it's essentially the same idea where the vessels now of the arms and legs are the ones that become occluded, which is blocked, or narrowed due to these sort of fatty deposits. Unlike patients who have myocardial infarctions or heart attacks, these PAD patients don't, it's not a fatality for them. Instead, the blockage of their arteries in their arms and legs will essentially cause pain while walking, pain at rest, and ultimately can lead to infections that become gangrene and ultimately need limb amputation. So for patients with PAD, the worst case scenario is a very severe form called critical limb ischemia that will ultimately require limb amputation. So as I talk about these two different diseases, heart attacks as well as PAD, a very important player in both of these diseases are endothelial cells. And that's really the focus of our talk today, because we really need to understand what endothelial cells do and don't do in order to treat atherosclerotic diseases. So endothelial cells are the cells that essentially line the inner layer of your blood vessels. And they play a very important role. So one, they are the cells that physically contact blood. So they are present in every single blood vessel in the body. And they're the ones that are physically contacting the blood. So you can imagine that endothelial cells play an important role because they're kind of like the gatekeeper. They are in part what controls proteins, what types of cells, what things go in and out of the bloodstream. And you can imagine that if your endothelial cells, for whatever reason, become sticky, and things like proteins, things like cholesterol want to stick onto those endothelial cells, that is essentially the beginning of atherosclerosis. So instead, the health of the endothelial cells is critically important. If the endothelial cells are healthy, they essentially make the blood vessel surface, inner surface, seem like Teflon. Like nothing wants to stick to Teflon. So the endothelial cells, when they're in a healthy form, will make your lining of your blood vessel feel like Teflon. But in other cases, when the endothelial cells are dysfunctional, they make your inner lining feel more like Velcro. And so you can imagine that in order to be able to control and treat atherosclerosis involves essentially understanding what causes the endothelium, which is essentially your layer of endothelial cells, what causes them to act like Velcro versus Teflon. And so one of the important clues about how and what determines endothelial function has been primarily the effect of the mechanical flow. And the reason is they experience blood flow all the time. And so you'd imagine that blood flow must be somehow regulating how endothelial cells function. And in fact, the field of flow and its role in endothelial cells, and specifically in the formation of atherosclerosis, has been really well studied in the past, and it's actually quite interesting in that in segments of your blood vessels that are really just straight and uniform, the flow there is what we call laminar flow. So it's a fairly uniform type of flow. It's very straight. It's very steady. But when your blood vessels actually bifurcate, or they turn, they bend, they branch off, or whatever, at those regions of your blood vessels, the flow there is what we call disturbed. So it's not really a uniform blood flow. And so you can sort of see that rather than having really uniform sort of flow fields, you sort of have these different recirculating eddies. So the flow there is really just not uniform. And so it's kind of interesting that in these different flow regimes, the way the endothelial cells look as well as function are also different. So in the area of laminar flow, so these are the healthy flow regions where the flow is very steady and uniform, those endothelial cells are happy. So they are kind of like the Teflon endothelial cells. They secrete a lot of molecules known as, one is nitric oxide, which is essentially like a Teflon agent. So in these straight segments, the endothelial cells are functionally like Teflon-ish endothelial cells. And interestingly, those endothelial cells have a specific look. So if you look at them, so in this case, the arrow is showing you the direction of blood flow, and the endothelial cells are actually are aligned. They're kind of really elongated. So rather than being a circle, they're really elongated along the direction of blood flow. So we call them aligned. But in these disturbed flow regions, like these bifurcations or these branch points, those endothelial cells are not happy. They are secreting all the molecules that make them into Velcro. And these endothelial cells also morphologically, meaning the way they look, is also kind of different. Rather than being sort of oriented longitudinally along the direction of blood flow, they kind of have a disorganized look to them. And so the documentation of how these endothelials function and how endothelial cells look with respect to different types of blood flow has been well described by Peter Davies, John Cook, as well as other people. But one of the questions that we as engineers ask is, well, the way that these endothelial cells are functioning, is it because of the actual flow that's causing them to behave that way? Or is it because of the fact that they are patterned in a certain way? And this is really a difficult question to tackle because the actual flow is doing both. So in the regions of laminar flow, the flow itself is actually causing the endothelial cells to pattern in this aligned way, whereas in the disturbed flow region, that type of flow field is actually causing the cells to pattern in a disorganized way. But we wanted sort of to dissect out these two different phenomena, and we essentially are wondering whether or not we could use a different approach to pattern endothelial cells in the absence of shear stress or of this laminar flow, and ask, can we use patterning alone? So if we could pattern cells in the absence of any type of shear stress, would the patterning alone dictate how the endothelial cells function? And there's a lot of reason for why we would be interested in that. So one of the translational or one of the medical applications has to do with the fact that atherosclerosis occurs at very distinctive locations in the body. So in this diagram showing essentially the aorta down here, the regions that are kind of darkened in the gray color are the places that tend to have atherosclerotic lesion formation. And those locations, as you can sort of notice, tend to be preferentially at these regions that are either a branch point or a curve or a bend or a branch. So, and because of that, that was also another motivational factor for us to wonder, well, if we could, for example, develop an artificial blood vessel that will always be happy, maybe that could be a really great medical product to treat patients who have clogged vessels. Give them an artificial blood vessel that essentially cannot be clogged. So let me tell you a little bit about how we are going about doing this. So the question that we essentially want to ask is, essentially, does the cellular orientation or their patterning by itself, can that induce the healthy endothelial cell characteristic or their function in the absence of blood flow? And again, in the absence of blood flow is important because typically when you give them blood flow, the flow itself is also imparting some type of patterning signal and cue. But if we can pattern cells using an approach that does not use flow, then we can sort of dissect apart the contributions that patterning does to endothelial cell behavior, as well as the role that the flow itself does on endothelial cell behavior. So we use an approach to essentially pattern endothelial cells by aligning them using nanopatterned biomaterials. So this is a biomaterial called collagen. It's a native biomaterial. It's found in blood vessels. It's found in pretty much almost all tissues of your body. But we use a very sophisticated extrusion approach where we can essentially create these nanoscale, as in like 30 nanometer thick fibrils. So think of like 30 nanometer spaghettis that are all aligned, in this case, in parallel. And what's really interesting is that endothelial cells will be able to sense this type of orientation. They'll be able to tell that, well, the underlying nanofibrils are all aligned in parallel, and actually they'll actually be guided by the direction of the nanofibrils and become aligned as well. So I'll show you that in a moment. But so these are scanning electron microscopy images showing these collagen nanofibrils where we've been able to essentially fabricate them where each fibril is aligned in parallel, or these collagen fibrils are all randomly oriented. And to put in perspective, you can imagine that an endothelial cell might be about 20 to 30 microns in diameter, and these fibrils are about 30 nanometers. So several orders of magnitude smaller than the size of a cell. So when we grow these endothelial cells on either this randomly oriented or this sort of parallel aligned scaffold, we can see that the cells actually look quite remarkably different. On the aligned collagen scaffold, the endothelial cells are actually, if you kind of look at this staining, which is F-actin. So F-actin is sort of a marker for the cellular skeleton. So if you can imagine a cell having a skeleton, this is what the F-actin is. And it's essentially showing you that endothelial cells are oriented along the direction of the collagen fibrils, which in this direction is in the horizontal direction. Whereas on sort of this randomly oriented, like spaghetti-like type of collagen scaffold, the endothelial cells are kind of just randomly distributed, just as is. And of course, you can do some further quantification of these observations. One simple one is by essentially measuring this, what we call their degree of alignment. So the degree of alignment is essentially what we call the angle that's formed by the direction of the cell relative to the direction of the collagen fibrils. So if a cell were perfectly aligned along the direction of these aligned collagen fibrils, they would have a degree of alignment of 0. So we can see that on the aligned collagen scaffolds, these endothelial cells are within 10 to 20 degrees from the axis of the collagen fibrils, which basically means they're pretty well aligned along the direction of the collagen fibrils. And this is really interesting because we were able to pattern these endothelial cells longitudinally as if they were in a healthy segment of the blood vessel, but without having to use any shear forces or fluid flow at all. And of course, it's not just looking at their cytoskeleton, which is again the cellular skeleton. There's also some of the efforts in our lab to actually look at some of the very basic signaling mechanisms that are mediating this. And one of the things that we're looking at are focal adhesions. So you can imagine the focal adhesions as being on the periphery of the cell. So you can think of them as little suction cups that a cell has. So if you can imagine a cell that has suction cups all over their body as a way to attach onto these scaffolds, that's what these focal adhesions are. And so we're noticing that not only is their actin cytoskeleton aligned along the direction of these collagen fibrils, but also these little suction cups, these focal adhesions, are also more organized as well. So they are actually organized along the front and the back ends of the cell, whereas on a randomly oriented collagen scaffold, those focal adhesions, which in this case, a specific focal adhesion is paxillin, you can see that these punctate green spots are pretty much distributed all around the cell. So it's great that we were able to get these endothelial cells to align as if they were in a healthy segment of a blood vessel that has sort of healthy laminar type of flow. But that's really all looks, isn't it? How do we know that when they are aligned, that they actually are functionally more healthy? So one of the assays that we use to study this is called a monocyte adhesion assay. So monocytes are cells that are floating within your bloodstream, and they are actually some of the first people to be involved in an atherosclerotic lesion formation. So the process of forming this fatty plaque or cholesterol plaque in your blood vessels is really a long, complicated process. But one of the very first events that happen is that inflammatory cells, which are what monocytes are, will actually attach to the lining of the endothelium. So that's what these are. These are endothelial cells. And different types of monocytes, leukocytes, other types of inflammatory cells will essentially first attach and then eventually build into this plaque. So monocyte adhesion is an early event that takes place in the process of atherosclerotic lesion formation. So we did an experiment where we essentially grew the endothelial cells on either the aligned collagen or on the randomly oriented collagen. And so as we would imagine, on the aligned collagen, the endothelial cells were aligned, and on the randomly oriented collagen scaffolds, the endothelial cells were randomly oriented. But then what we did was that we essentially then gave them monocytes. So what you're seeing here in these pictures are monocytes. So they were fluorescently labeled and took a picture under a fluorescent microscope to specifically look at these fluorescently labeled monocytes, which is why you don't see the endothelial cells. And essentially asked, well, would the aligned endothelial cells attach to monocytes in the same avidity or the same quantity as the endothelial cells that are growing on your randomly oriented control scaffold? And and as sort of maybe as we expected, what we found was that on the aligned endothelial cells, sorry, that's what the FD30 means, which is arbor diameter of 30 nanometers. On the aligned endothelial cell, there was substantially less monocyte adhesion, something about 50% less monocyte adhesion compared to when the endothelial cells are randomly oriented. And of course, we can do other types of assays. We did other types of confirmatory assays using ICAM1 as another marker of inflammation, and also showed in a similar way ICAM1 expression, which is another inflammation-associated molecule, was also reduced when the endothelial cells were aligned. So we thought this was really exciting data, because what it's telling us is that even if we didn't have any flow involved in the process, by virtue of just patterning the endothelial cells, in this case using scaffolds, we could essentially modify both their orientation as well as their function. And so a light bulb then hit. Okay, well that's a great biological finding, so how can we use this to help therapeutically? And so the idea that we came across is like, well, we know endothelial cells are very important, and from our data it seems as if we could pattern the endothelial cells, they'll remain happy. So one of the knowledge that we know about is that for patients who have atherosclerotic lesions in their heart or other places, a common procedure that they'll have to mitigate the problem is to have a bypass graft. So essentially it's as if the highway is blocked, and the bypass graft is the detour vessel that's allowing blood flow to go to this detour bypass graft so that you're able to have blood flow through your heart or through whichever part of the body it is. And we know also scientifically that these bypass grafts that people put in don't work that well. And part of it is because these regions that require the bypass graft are the regions that experience disturbed flow in the first place. So if you stick a vascular graft as a bypass graft into an area that has disturbed flow, the disturbed flow being an unhealthy flow regime may actually make those endothelial cells in the vessel unhealthy, and then you might have another atherosclerotic lesion formation again. So our idea was, well, if we could essentially create a synthetic vascular graft where we're essentially forcing the endothelial cells to align longitudinally along the direction of blood flow, which is sort of mimicking how healthy endothelial cells should look like, such as here, then maybe even if we implant this bypass graft into an area of very unhealthy disturbed flow, those endothelial cells, because they see this nanopatterning, may stay healthy anyway. So that was our big idea. And so what we did was we developed a version of the collagen scaffold that is essentially a graft. So it's a hollow cylindrical graft, which we can then essentially seed endothelial cells inside the graft and then implant them into animals to see whether or not the aligned nanotopography actually helps to improve the ability of these vascular grafts to not get blocked or occluded. So, and those studies are definitely ongoing, and it's definitely an area of our interest. And so one of the questions you may ask is, okay, if you want to make these grafts that go into people, are they really strong enough to be able to even go into people in the first place? So one of the characterizations that we do is the mechanical characterization. And some of the properties that we measure is the graft's elastic modulus. So it's essentially a measure of how stiff or how flexible, you may want to think of it, the graft is. And its burst pressure. Is it able to withstand physiological blood pressure, or will it just burst if you try to put it into your own blood pressure? So we do these measurements, and these grafts do have a reasonable elastic modulus as well as a burst pressure that would withstand human physiological conditions. And we think, that's great. But one of the questions that we haven't answered is, okay, we know that in the absence of any type of flow, by virtually patterning them alone, the endothelial cells are happy. But if you were to then subject them to an unhealthy, disturbed flow, would they still remain healthy? And ultimately, if we want to implant them into the body where these bypass grafts would be in regions of unhealthy flow, that this is a very important question that we have to address. And so to address this question, what we essentially did was we grew the endothelial cells on these aligned nanofibrillar scaffolds, and now they're aligned as if they were happy, but then subjected them to disturbed flow, which is unhappy. And the question is, who wins out? Do the cells stay happy because they are patterned, or do they turn unhappy because now they're experiencing an unhealthy flow? So to do this, we collaborated with two colleagues of ours in chemical engineering, Alex Dunn and Jerry Fuller, and we, who actually were the ones who initially developed this specific impinging jet device. So it's a device where essentially allows you to give disturbed flow, as well as be able to image it microscopically at the same time. So in this setup, what you're seeing here is a dish of cells, and in this case, this will be our cells that are seated on the aligned collagen scaffold. And then there's actually a jet that is essentially dispensing disturbed flow onto the surface of this dish. So it's actually just hitting the cell like a faucet with disturbed flow. And then underneath the dish is actually a microscope that's recording it in real time for 24 hours. And you can imagine that, so this is to give you an idea of how the setup looks. Our jet is actually is a circular jet, and so when we grow our endothelial cells on the aligned collagen scaffold, this is just a schematic. You can imagine that in green are your aligned endothelial cells that are growing on the aligned collagen scaffold, or in this case, on the randomly oriented collagen scaffold, the endothelial cells are randomly distributed. But what you're seeing here in the circle, this is the region of the jet. So the jet is hitting this region with disturbed flow. And the flow is actually radial, so it hits the cells and then it just flows out radially. So that's why the arrows point out radially. And so we've done finite element modeling to model the mechanics of the fluid flow, and it does mimic sort of what we would see, for the most part, in physiological conditions of blood flow, where we have these sort of recirculating eddies, which are characteristic of disturbed flow. And so then what are we actually studying? We're actually studying a number of different things, but one of the things that we were most interested in was if you put the aligned endothelial cells in the presence of disturbed flow, do they even stay aligned in the first place? So the one sentence summary is yes, they do. They actually remain aligned along the direction of the scaffold, even when they are exposed to disturbed flow. So this is just some data from the work of my postdoc, in which case I'll be talking about a lot of her work for the rest of this talk, show that even the presence of this unhealthy disturbed flow, the endothelial cells essentially stay aligned within 20 degrees from the axis of the collagen fibrils. But what I thought would be actually more interesting for this audience is actually show you some of the movies. So these are some of the time-lapse movies. So we actually took essentially still images every 20 minutes over the course of 24 hours to track what's going on with the cells. How do they migrate? And then ultimately, what orientation are the cells at the end of 24 hours? So what you're seeing here is, so this is essentially the region of where the jet is essentially jetting media onto this dish. And what you don't see very clearly here is that these endothelial cells are growing on top of the randomly oriented collagen scaffold. And so this is sort of how the movie would look if you sort of stitched the stills at every 20 minutes. So you want to focus on what's going on in here. So you can sort of play it a few times because it's kind of hard to follow each and every cell. But you can sort of see that the endothelial cells are kind of, some of the cells are migrating in, some of them are moving in lots of different ways. But then at the end of the movie, what you really see is that the endothelial cells, particularly under the jet and sort of surrounding the jet, are pretty much randomly oriented. And we can do further quantification of that. So one is that we can actually essentially follow each cell, in what we call cell migration trajectory movies, and give each cell a color and essentially follow each cell over the course of those 24 hours. So we can see, in this case, the cells are really being able to migrate outwards. And in this case, we're actually just following cells at specific regions at 3 o'clock, 6 o'clock, 9 o'clock, and 12 o'clock. But they're able to just migrate radially out. Now, in contrast, these are endothelial cells that are growing on the aligned collagen scaffold. So you can sort of see, so here you can actually see the aligned collagen scaffolds in these sort of strips that are oriented horizontally. And so I want you to sort of pay attention to what's going on to endothelial cells sort of in this region, which is where the jet is. And so if you look at this movie, you can sort of see that the endothelial cells are some of the cells are migrating inwards towards the jet. But for the most part, if you look at sort of the last still, most of the endothelial cells are pretty much aligned along the direction of the collagen fibrils, which is, in this case, a horizontal direction. And I'll play that movie again so that you can sort of see the cells migrating in, but then they pretty much still stay horizontally oriented, which is along the direction of the collagen fibrils. And of course, you could then do trajectory analysis and follow each particular endothelial cells. So this is just a subset of some cells that we followed over the course of time. And what's interesting is, the reason why we're interested in specifically sort of like 3 o'clock, 6 o'clock, 9 o'clock, and 12 o'clock is that at 3 o'clock and 6 o'clock, being that the flow is radial, at 3 and 9 o'clock, the flow is actually, in this case, parallel to the direction of the collagen fibrils, as well as parallel to the direction of flow. So they're both sort of aligned. In contrast, at 12 o'clock and at 6 o'clock, they actually are perpendicular. So the flow is in this direction, is heading vertically, but the collagen fibrils are still oriented horizontally. And so the question is, what's going on at 12 o'clock and 6 o'clock? Because in those regions, the flow is perpendicular to the direction of the cells. So let me play this trajectory again, so you can take a look at, maybe if you want to compare, I don't know, like 3 o'clock versus 12 o'clock. So let's look at how they respond again. So what you're basically seeing is that at 3 and 9 o'clock, so again here, the flow direction and the collagen fibril direction are in parallel. The cells are migrating in, and they're migrating, maybe on your screen it might look like about an inch. But at 12 o'clock and at 6 o'clock, those endothelial cells aren't really migrating much, right? They're kind of actually just just stay there, they move a little bit back and forth, but they don't really migrate as much. So what it's showing is that the endothelial cells at 12 o'clock and 6 o'clock, so again, those are the areas where the flow is perpendicular to the direction of the cell orientation. They pretty much stay over there. Whereas at 3 o'clock and 9 o'clock, where the flow direction and the collagen fibril direction are both in sync, the endothelial cells continue to migrate along the direction of the collagen fibrils, and they migrate faster. So this is actually a very interesting data piece, because it's telling us that even if we subject these aligned endothelial cells to flow in the perpendicular direction, which is 12 o'clock and 6 o'clock, those cells still stay aligned. But we also notice that they don't migrate as much compared to the cells at 3 o'clock and 9 o'clock, where the flow and the collagen fibrils are in sync. So this is definitely, some of the research areas that we're interested in. And you may wonder, okay, so now if you were to do the same monocyte adhesion experiment, what would you see? So we did the experiment where we had the endothelial cells either on the aligned or the randomly oriented collagen, and we exposed them to the disturbed flow using the jet, and then afterwards we exposed them to these fluorescently labeled monocytes, which are what you see up here. And essentially what you see is that on the aligned collagen sample, you see a lot less monocyte adhesion compared to on the randomly oriented collagen. So again, it's showing us that when the endothelial cells are aligned along the direction of the collagen fibrils, they stay happy even if you give them an unhealthy disturbed flow, because of the fact that they're not really adhering to monocytes as much. And so we think this is really exciting. So it's giving us, I guess, further evidence that if we were to develop an aligned vascular graft, they actually may be quite therapeutic in being able to stay as a bypass graft in the body that does not get occluded or blocked even in the presence of disturbed flow. And so one of the obvious next steps is that we are in the process of doing some more in vivo testing. And in addition to that, we're also doing some second generation designs of vascular grafts, which is, besides endothelial cells, which are important, shown in the green, blood vessels actually have multiple concentric layers. And a very important concentric layer are the smooth muscle cells. So the smooth muscle cells in this diagram of a native vessel, where the endothelial cells are shown in white, the smooth muscle cells are the ones in blue. They surround the endothelial cells. And interestingly, their cellular orientation is circumferential. So rather than being aligned along the direction of endothelial cells, which is longitudinal, they're circumferential to that of the endothelial cells. So we can sort of apply a similar type of methodology to pattern the smooth muscle cells in a circumferential orientation as well. And so this is some of the ongoing work that both Karina and Prajakta are working on together as well. So I think, I'd like to end the talk here so that we could have room for discussion and questions. But this is just to give you a little bit of a snippet of some of the work that we do to apply bioengineering as well as science principles to study fundamental biology questions, and then try to translate that knowledge into something that has a translational or medical treatment purpose. So thank you for this talk, and I'm happy to take questions and acknowledge members of my lab. Thank you. Yes.
So, have you put these cells in a 3D printer yet?
We have not put them in a 3D printer yet. For a couple of technical reasons, the 3D printers even though 3D printers are a lot more affordable as well as much better than the past few decades, they have a limit of resolution still in the order of like 100 microns. So it's very difficult for them to, difficult for us to create 3D scaffolds with sort of features that are in the nanoscale at this very particular moment. And I think that in the near future, when the technology improves, we may be able to use just 3D printing to create these nano-patterned scaffolds. Yes?
You said earlier that the hands are really clinic too, and do you think that that's going to work?
Sure. So I mentioned sort of the general areas where bypass grafts are most often used. I think that in terms of where the greatest need is right now, I think that the existing bypass grafts that are commercially available that are for use in large vessels, such as in the aorta, the coronary arteries, those right now commercially are primarily just polymer-based grafts, so they don't have any cells on them. But they've been working pretty generally well as in what we call being very patent, meaning not being occluded for about like 5 to 10 years at the very least. But the problem area, which is also the area that I see great opportunity for cell-based biological vascular grafts, is in the small diameter vascular grafts. So the small diameter vessels that get occluded are the ones that usually are, let's say, in your arms and legs. So particularly for PAD patients, the vessels that get occluded are primarily the ones that are below the knee. So those ones actually are very difficult to replace with an artificial bypass graft right now because they are very small, as well as they have very low flows. So these two things combined is a death sentence for these grafts. So they get occluded really quickly. So we think that these cell-based vascular grafts have greater opportunity in these small diameter vascular graft applications. How big? Sure. So starting from something that's research-based, we're at the point now of doing, small animal studies, large animal studies, clinical trials. So we're talking probably about minimum 5 to 10 years, and as long as 20 to 30 years. Any further questions?
Instead of using printing or whatever, have you evidence of using vapor-based deposition?
Yes. So we haven't looked at that one, the vapor deposition approach very much, only because of the fact that we want to use, the simplest approach. And so far, the way we engineer these collagen scaffolds are super easy. In fact, you pretty much only need a syringe and a needle and some saline to be able to, and as well as collagen, to be able to create this nanotubullar structure. So while there are more sophisticated technologies like 3D printing, like vapor deposition, and we can think of a whole bunch of other techniques, this approach has been just a lot cheaper and easier to do without sophisticated technology. But that there are definitely multiple ways to make it.
Let's give her a hand. She will probably stay around.